So in Mendelian Inheritance Part 2, Episode 2, we're going to be looking at mitosis. Now here I have a picture of mitosis. A good thing to know is that interphase is not included in mitosis. It's more of a preparatory phase. So is cytokinesis. It's like kind of the ending phase. Some textbooks will consider cytokinesis on its own, not as a part of mitosis. So if a test asks you what are the four phases, say prophase, metaphase, anaphase, telophase. If they ask you what are the five phases, then you can include prophase, prometaphase, metaphase, anaphase. Prometaphase is really just late prophase. But if they ask you just to name four, then don't include prometaphase. If they ask you to name five, then include prometaphase. And once again, interphase and cytokinesis are not necessarily considered to be part of this process. So I'm just going to be reading off key terms because I'm going to be using these terms a lot throughout the video, so it would be good if you're familiar with these. So centrosomes are where the microtubules elongate from. So inside the centrosomes you have centrioles, and then the microtubules are polymers of tubulin that are the pathways for motor proteins across the cell. And then a kinetochore is essentially a protein complex in which microtubules/spindles attach to. And then centromere is a DNA complex that is in the middle of the kinetochores and the center of the chromosome. So a good thing to know is that a lot of people get kinetochore and centromere confused. So there's not really any way that I remember. It's just like I just read it. I just read a lot of textbooks, so like this stuff is like easily memorized. But just know that kinetochore is just a protein complex, and then centrosome is a DNA complex that's in the middle. That's all you need to know. Those are the two differences. So chromosome is is just like genetic information in a thread-like structure, and then there's one centromere per chromosome. So you can count the number of chromosomes by the number of centromeres, and then sister chromatids are the two units of chromosomes that share the same centromere. So those are the terms you should probably be familiar with. Now let's talk about the types of microtubules. First one is we have kinetochore microtubules. So they're embedded in a protein complex called the kinetochore, as we said, and they assemble at the centromere of each chromatid. So the kinetochore microtubules are responsible for chromosome movement during cell division. Non-kinetochore microtubules extend towards each other from two polar centromeres and overlap to elongate and stabilize during cell division. And then astromicrotubules grow towards the membrane of the cell, where they attach and contribute to cell stability. So now that we have all the terms out of the way, let's actually start, narrating and talking about mitosis. So the interphase, like I said, it's more of a preparatory phase. So it's broken up to actually three phases. Don't worry, G0. I'll get to G0 in a second. But the first phase in interphase is called the gap synthesis phase. And so essentially, in gap synthesis, gene expression is really high. So gene expression is like protein synthesis and transcription. So these processes are really high and very active. And so then and essentially in G1, the cell is growing to about double its size, and then more organelles are produced, and the volume of the cytoplasm increases. So just really know the two important things to know is that gene expression is high, and the cell increases in size by roughly about double its previous size. So the synthesis phase is essentially when DNA is produced by the synthesis phase is the production of DNA by semi-conservative DNA replication. So what you need to know is that semi-conservative DNA replication is essentially when the DNA strand that's being produced is consisted of one parent strand and one daughter strand. Don't worry too much about DNA replication. I had a video on DNA replication, but I'll probably do a more intricate video on DNA replication. But just know that DNA is produced. Don't worry about the semi-conservative DNA replication just yet. So G2 or gap 2 phase, the cells continue to grow and the mitochondria divide, And in plants, the chloroplasts also divide. So this is the last phase before the M phase, also known as mitosis. So really, it's just getting prepared for the mitosis phase. And then the G0 phase is an extended or permanent G1 phase in mode the cells no longer divide. So the cells enter a state of no longer dividing. And an example of this is neural cells in mammals. So here you can look at the pictures. Okay. So the prophase is essentially chromosome condensation. So the genetic information in chromosomes begin to condense and making the chromosomes visible under a microscope, and then the centrosomes begin to migrate to the opposite poles by extending their microtubules to make an early meiotic spindle. The nucleolus also disappears. So key things to know is that nucleolus disappears. It's the formation of an early meiotic spindle. Centrosomes begin to migrate to the opposite poles, and genetic information in chromosomes condense and make it visible under a microscope. So prometaphase is the nuclear membrane / envelope disappear. So having reached opposite poles of the cells, the centrosomes extend microtubules that attach to the kinetochores of the chromosome centromeres. So microtubules extending from the opposite poles exert pulling force in both directions. Chromosomes move towards the middle of the cell, and then cohesion binds sister chromatids to resist premature separation due to pulling forces. So cohesion is essentially just a protein that binds to sister chromatids. And then non-kinetochore and astral microtubules stabilize the cell. So I would say the most important thing to know for prometaphase is that just the nuclear membrane disappears, and then the microtubules start to extend from the centrosomes, because the centrosomes have already migrated from opposite sides of the cell. And so metaphase is essentially complete chromosome condensation is reached in the metaphase. So the chromosomes have fully condensed in this part, and then the chromosomes begin to align so that the sister chromosomes of each chromosome lie on the either side of the metaphase plate. So essentially, we just have the alignment of chromosomes and daughter chromatids in the metaphase plate or in the middle. In this phase, you can take a picture of the chromosome, so karyotypes are also produced in this phase. I forgot to add that. And then the sister chromosomes of each chromosomes are attached to the kinetochore. So the sister chromatids bind to the kinetochore or bind to the protein complex, because we said kinetochores were a protein complex. And then kinetochore, non-kinetochore, and astral microtubules are fully extended from the centrosomes, and complete meiotic spindle is in place. So the meiotic spindle is now formed. So really what you need to know is that it's just the alignment of chromosomes / daughter chromatids in the metaphase plate, and then the meiotic spindle is finally formed, and the chromatids bind to the kinetochore or the protein complex. That's all you need to know for metaphase. Then anaphase, the sister chromatid separation or disjunction, the separation between the chromatids, occurs through the breakdowns of sister chromatid cohesion. So remember when we said the protein cohesion helps bind the sister chromatids? Well, now in anaphase, that cohesion or that force is broken down, so the depolymerization of kinetochore microtubules helps separate these daughter chromatids away from each other. So polymerization of kinetochores polymerization of non-kinetochore microtubules accompanies the movement of daughter chromosomes, giving a cell an oblong shape at the end of anaphase. So the cell takes a more more an ellipse, more of an ellipse shape. So key thing you just need to know for anaphase is just that they just separate, and they just separate. That's it. That's literally it. They just separate, and the force from cohesion gets broken. And cohesion is a protein. So telophase and cytokinesis is very simple. Telophase is separated from cytokinesis. So in telophase, non-kinetochore microtubule polymerizations continue to elongate during the cell, pushing the poles apart. The nuclear envelope begins to reassemble and will shortly surround the chromosomes. Chromosome decondensation accompanies during the nuclear envelope reassembly. So what you need to know is that two key things happen in telophase is that the nuclear membrane / envelope reappear, and then the chromosome decondense in order to be transcribed in other molecular processes such as gene expression. And then cytokinesis is actually the division of the cytoplasm, and so this helps create two new cells with separate nuclei, or the formation of new cell walls in plant cells. And then, yeah, that's all you need to know. And then you need to also know this is that the nucleolus reforms. So remember when we said, essentially, so what you need to know is telophase and cytokinesis are kind of the reverse of the prophase, because in prophase, the nuclear membrane dissolves, but in telophase and cytokinesis, they appear again, and then the nucleolus dissolves in prophase, and then they reappear again in telophase. So a good way to memorize telophase is just to know that it's the reverse of prophase. And yeah, that is the end of the video. Thank you for watching.